Hi guys, my name is Jingyin and I'm a PhD student from the University of Queensland. And today I'm going to talk about developing cryopreservation for endangered Gossia, a genus plant threatened by Myrtle Rust. And I would also like to start by acknowledging the country on which we meet here today and pay my respect to the elders, past, present, and emerging. So I'll first give a brief introduction to the genus Gossia. Gossia belongs to the Myrtaceae family and it includes 47 accepted species and they all distribute in Australasia. And it is also the main genus in Myrtaceae that has been reported to have these plant hyperaccumulators, which potentially can be used in phytomining and phytoremediation. And many Gossia species are now threatened by habitat loss, weed invasion, and Myrtle Rust. A majority of Gossia species are susceptible to Myrtle Rust, while for the impacts of the disease on specific Gossia species may differ according to their susceptibility and conservation status. And as mentioned earlier, there are several Myrtaceae species has been classified as Category X and Category Y species according to if they will decline within one generation due to Myrtle Rust. And there are 4 Gossia species, including Gossia helii, has been included in Category X, and then another 7 Gossia species has been included in Category Y. And currently, there are 2 Gossia species, Gossia fragrantissima and Gossia gonoclada are listed as endangered according to the EPBC Act, and these 2 species are also a focus of my current research. And the 2 species are also included in the very high priority species in the Myrtaceae National Recovery Action. And my PhD is also founded by Logan City Council, and they wanted me to help them develop a cryopreservation protocol for them to conserve the remaining genetic diversity of Gossia gonoclada in the wild. So in terms of conservation options for threatened plant species, there are 2 types of conservation, in situ conservation and ex situ conservation. So in situ conservation refers to the preservation of plant species and its associated ecosystems at the natural habitats. So an advantage of this method is that it allows the plant species to co-evolve with the surrounding ecosystems and therefore makes it more adaptive to ecological changes. But because these plant species are conserved in situ, it is very hard to manage and avoid these environmental risks such as Myrtle Rust. And a good alternative for in situ conservation is ex situ conservation. So it refers to preservation of plant species away from the natural habitats. And the common ex situ methods include seed banks, living collections, tissue culture, and cryopreservation. And so I will mainly focus on the latter 2 in vitro method in my research. So for tissue culture, it is normally known as a way to propagate plant species, but it can also be used in a method of conservation. But due to the cost in labor and the space to store the in vitro cultures, it is normally used as a short-term conservation strategy. And tissue culture normally consists of 4 stages: culture establishment, multiplication, rooting, and acclimatization. So as mentioned by Lindo earlier, my collaborators at Australian Plant Bank has also been working on the conservation of Myrtaceae species, and they have successfully developed tissue culture systems for Gossia fragrantissima. And using their protocol, I was also able to establish some of the Gossia in vitro cultures in our lab. Using the plants that was given from another research group, and it was initially tagged as Gossia fragrantissima, but later we found out it was not the species, and we will need to maybe use sequencing and some other method to identify its identity. And currently, I'm also working on developing tissue culture system for Gossia gonoclada. I was able to get some cultures for initiation, but still working on modifying the media for multiplication. So it is meaningless if we can't transfer these in vitro derived plants into the natural environment. And I've also successfully rooted and acclimatized some of these in vitro Gossia plants, and we hope to talk to Logan City Council to using these in vitro derived plants to help the restoration of Gossia in the wild. And cryopreservation is a long-term storage of regenerable plant tissues in ultra-low temperature, usually in liquid nitrogen. So the biggest difficulty in developing a cryopreservation protocol is to avoid cryo-injury, which are normally caused by the formation of ice crystals. So to do that, we will need to induce vitrification of cellular moist instead of crystallization. And there are many different cryopreservation techniques, but the one I used here is droplet vitrification, and it is one of the most commonly used methods in plant cryopreservation and has already successfully applied on thousands of plant species. And on the right here is a diagram of some major steps in a droplet vitrification protocol. So the first step is to obtain some donor plants, and then the explant will be dissected from the donor plants. After that, they will be treated with loading solution for osmotic protection and then dehydrated in vitrification solutions. And after that, they will be placed onto alpha strips in drops of vitrification solutions before they get quickly plunged in liquid nitrogen for storage. And if we want to regenerate these explants, we need to pour these out and put them in unloading solution for rewarm and rehydration and regenerate them in vitro cultures. So currently, I'm mainly working on developing cryopreservation for Gossia fragrantissima, and according to some preliminary experiment, we found that the shoe tips of Gossia fragrantissima were very difficult to survive after being exposed to liquid nitrogen. And in the next few slides, I will try to show you how I did to optimize the protocol. So the first thing that I looked at is explant type. So there are a series of different explants that we can use in plant cryopreservation, including seeds, embryos, callas, and shoe tips. So I've used auxiliary buds and apical shoe tips here. The first reason is that I can only access to these types of material, and also in terms of conserving genetic diversity, it is usually very beneficial to have these types of explants because they will less likely to have small clonal variation. So after comparing the viability of the shoe tips after dissection, after treated with loading solutions and after treated with vitrification solutions, I've chosen the apical shoe tips as explant to continue the next experiment. This is not only because they had overall better viability compared to auxiliary buds, but they are also more uniform in size and structure, which might be beneficial in developing cryo protocol because they will have consistent response to cryopreservation process. And here's a photo showing you some dissection control shoe tips from week 1 to week 4. And as you can see, they are very happily lived. But when these shoe tips were treated with loading solutions and PVS2, a lot of the shoe tips lost viability and died, and because of the toxicity of vitrification solutions. So in the next experiment, I was trying to incorporate donor plant pretreatment into the protocol to see if I can induce a better tolerance of PVS2. So in this photo here, there's some visual examples of survival and regrowth shoe tips. So this is the criteria I used to score the shoe tips. So let's look at the first 4 sets of data here. The black bar represents survival and the gray bar represents regrowth. So as we can see here, there's no significant difference between shoe tips dissected from fresh cultures and cold pretreated cultures, no matter treated with loading solutions for 10 minutes or 20 minutes. But after the shoe tips being treated with loading PVS2 solutions, for fresh cultures, it seemed that loading 20 minutes supported a better regrowth compared to loading 10 minutes, but the same effect were not observed on shoe tips dissected from cold-treated cultures. And it could be because the cold treatment has induced better tolerance of PVS2 by reducing the required duration of loading treatment. So in summary, I've still been working on developing tissue culture systems for Gossia species and has successfully acclimatized some of these in vitro cultures. And I'm still working on optimizing the cryopreservation protocols. And I'm also going to study the post-cryo survival and regrowth once I got ideal survival before the liquid nitrogen exposure. And I also hope to, if I got the protocol done, I hope to transfer it to other Gossia species and a wider Myrtaceae species under threat. So I would like to thank for the fundings and plant materials from these organizations and also the travel bursaries from Australasian Plant Pathology Society for me to attending this conference. Thank you.
Probably a stupid one, but can you, just something we were talking about earlier, can you tissue culture like roots, and can you tissue culture, like root tips, and then potentially have like mycorrhiza, unculturable fungi conserved that way?
Yeah, it is possible to tissue culture roots, but I've never tried it myself, so it's really hard to answer that question.